Hello and welcome back to the video. Till now we have been discussing about the transcription only. In the first part of the transcription, we have discussed how the transcription in prokaryotes took place, second about the eukaryotes, and third part was regarding the modification, which we discussed about the polyadenylation and the capping process. In the last video, we have discussed another part of a modification, that is splicing, which we have discussed half, and rest half we will be discussing in this part. So let's begin. Before that, I am going to give a quick recap which we have already discussed in the last video. So in the last video, we have discussed that what is splicing. In short, we can say splicing is the removal of the introns. Introns are the non-coding part which do not take part in the protein synthesis. That's why they are needed to be removed out. And the introns removal, or you can say the splicing, are of different type. First is exon skipping, second is cryptic splicing and signals, third is alternative splicing, fourth is trans splicing, okay, which we have already discussed in the last part of the video in detail. If you have not watched that part, to watch that part and get clarified each one of them in detail. If you have still doubt in this process, you can comment down. Okay. Next, moving the splicing method. Splicing methods also I have discussed in the last part of the video, that the intron, the branch point which have a 2' OH end, forms a nucleophilic attack on the 5' splice site. 5' splice site then gets detached, a phosphodiester bond is broken down, and OH which is formed in the 5' region forms the 3' splice site. It is attack or nucleophilic attack is done, and at the end of the result the intron is removed, and you can see intron is removed and the spliced exons are joined. Okay, so this is the method which we have discussed in detail about the last part of the video. In this part of the video, we are going to discuss certain 3 important mechanisms which the splicing undergoes. First is the pre-mRNA spliceosome splicing. This pre-mRNA spliceosome splicing is performed by a group of RNA molecule. Okay, this is very important. This mechanism is very important regarding exam point of view. It is asked in exam, and also the sequence of the RNA molecule which is used is also asked. So please do listen to it carefully. If any doubt, do comment down at the end of the video. So first what happens, so let us understand the each one step in detail. First what happens, what is pre-mRNA spliceosome splicing? It is performed by the RNA molecule. Okay, so the RNA molecule, what type of RNA molecule? Short 5, U1, U2, U3, U4, U5 and U6, known as snRNA. So 5 short RNA, snRNA means short nucleotide RNA, 5 short nucleotide RNA, that is U1, U2, U4, U5 and U6. This group of complexes, or you can say the RNA molecule, perform this splicing mechanism and form snRNP, that is small nucleotide processing RNP. Okay. Next, the ATP hydrolysis is not required in the RNA splicing. Okay, while the splicing process is being done, the ATP is not used, but for the arrangement of the complexes, this ATP is required. Splicing removal process energy is not required, but for the arrangement purpose, the energy, that is ATP, is used. First, let's understand this figure, then we will understand the process in detail. So as I have already told you, this is a pre-mRNA. Okay, this is the 5' end, you can see, and this is the 3' end. Okay, this is exon 1, this is exon 2, this yellow part is totally intron, and A, that is the adenine, here it is the branch point. Okay, and this site is known as 5' splice site, and this site is known as the 3' splice site. Apart from this snRNA, that is U1, U2, U4, U5, U6, there is certain other 2 proteins which are involved in this process. First is known as BBP. What is BBP? That is branch point binding protein. As I have already told you, this is the branch point that of the intron, that is adenine residue. For binding to this branch point, a protein is required, that is known as BBP. Okay. Next, another protein or factor is required for recognizing the branch point, that is known as U2AF, that is U2 auxiliary factor is used to recognize this branch point, and then after recognition the BBP protein will bind to it. Okay, you must be wondering why this branch point is necessary to understand, because the branch point is the site where the intron is present. Okay, so we need to remove this intron. So it is nearly clearly you should understand that in this process intron is needed to be removed. So for intron recognition purpose, there is a branch point is present in the intron. So proteins like the BBP and the U2AF bind to that region and gives a signal to this snRNA that I have come here and now you can come and bind and cleave the intron and remove the intron. That's why this branching point as well as the removal of this intron is possible because of these two factors, that is BBP and U2AF. Hope you are clear with this. Next, moving to the sequence. So first what happens, U1 snRNP, that is this one, UN snRNP comes and bind to the 5' splice site region. Okay. Next what happens, once this snRNP is bind to this structure, what happens, the BBP and the U2AF is removed, because the BBP and U2AF only function is to recognize the branch point region. Once the branch point is recognized or binded, it is recognized, that is no use of them. So they are removed. Then next factor, that is U2 snRNP comes and bind to this branch point. After binding to this branch point, this 2 will be removed, because the main purpose of this 2 is that so that this snRNP comes and bind here and the process of splicing will be done, because once this that is the snRNP bind to it, the rest of the function is done and the splicing is done. Okay. So for recognition purpose only, the BBP and U2AF is bind. Once U2 snRNP comes and bind to this splice, that is the branching point, there is no use of this BBP and U2AF, they are removed. Okay. Hope you are clear with this step. Next comes the other three, that is rest. First comes the U2 which binds U1 which comes and bind to the 5' splice site. Second comes the U2 which binds to the what region, that is the branching point, and then comes the U4, then U6 and then U5. Okay, U4, U6 and U5, these are known as triple snRNP which enters the reaction. Okay, they will enter now. What happens, the U4 and U6 snRNA are held firmly and the base pairing is seen. So the very much interaction, that is strong bonding between U4 and U6 will be seen, and their pairing will be seen between U4 and U6. You can see here now by this time all the four, that is all the enzymes are bind. After the U4 and U6 binding, U1 and U4 is removed. Okay. So rearrangement, break apart U4 and U6 base pair, following U6 to displace U1 at the 5' splice zone. So what happens in this region, after the U4, U6 and U5 comes, U4 and U6 binding takes place strongly. Okay, you can see. Okay, so U4 and U6 binding takes place strongly. Then what happens in the next step, due to the rearrangement of these U4 and U6, the U4 is removed out and U1 is along with that is also removed out, and lariat formation and 5' splice site cleavage will take place due to the removal of this U1, because U1 was present in this site, that is the 5' splice site. Due to the removal of this region, what will happen, a lariat formation will take place. You can see here lariat formation will take place. You can see, okay, the lariat formation and the cleavage will be formed. Now what happens? This 3' OH region of the 5' end, this region will form a nucleophilic attack on this 3' splice site. Okay. As a result of which, from here the intron will be released out. Okay. From here the intron will be released out. Now the intron is free. Now the intron is free and exons are joined. Once again I am trying to explain. First what happens? U1 snRNP comes and bind to the 5' splice site. Okay, 5' splice site it comes and bind, and then then BPB and the U2FA recognize this branch point. After recognition, the U2 snRNP comes and bind to this branch point. After that, these two are removed, BPP and U2A. Then comes the triple snRNP, that is U4, U6 and U5. Then there is certain rearrangement between U4 and U6, as a result of which the U4 is removed out. Along with that, the U1 is also removed out. As a result of which, what happens? This forms the breaks this bond, as a result of which the 3' OH end of this 5' end will form a nucleophilic attack on this 3' splice site, as a result of which this intron, this yellow portion, completely is removed out and the exons are joined by them. So this process is completely done by this 5 types of short RNA. Okay, this process is known as pre-mRNA spliceosome splicing. Hope you are clear with this. Moving to the cell splicing technique. So by now you have been seen that in this technique a special RNA is required for undergoing the splicing mechanism. But in cell splicing, the self structure, this intron itself is responsible, or certain internal structure is responsible for the splicing process. There are two types of cell splicing, that is group 1 and group 2. Before that, let me show you something. What happens in the pre-mRNA spliceosome? The intron which is formed is in the form of a lariat formation, like this. Okay. As well as the group 2 cell splicing, the intron which is removed is in the lariat form. You can see lariat form. Okay. Whereas in the group 1 cell splicing, the intron which is removed is in the form of a linear intron. This difference is mainly asked in exam between the group 1 and group 2 cell splicing. Remember, group 1 cell splicing, the introns which is removed is in the linear form, whereas the group 2 is in the lariat form. Apart from that, there are certain other differences also we will be dealing, but this major difference needed to be understood. So first let's discuss about the group 1 cell splicing. As I have already told you, this is the 5' end, this is the 3' end of the pre-mRNA. Okay, next this is the 3' exon, this is the 5' exon, this is the intron sequence, and remember here that here the branch point is guanine. Remember, previously what we were discussing, the branch point was adenine, but here guanine is the consist of 3 OH end and is the branch point. This is the major difference. Okay, please note it down. Okay, what happens? This G, that is guanine, have OH which forms a nucleophilic attack on this 5' splice site, as a result of which the phosphodiester bond between these two will be removed and this free OH will be formed. Again this OH will form a nucleophilic attack on the 3' splice site, and then what will happen? The intron will be removed and the intron is in the form of a linear shape. This is very much important. The intron is in the linear shaped. Okay, sorry. The intron is in the linear shape. This is very much important to be understood. Okay. Next, the exons are joined. Okay. The 3' OH of a free guanine nucleotide located in the intron. Here you must remember that the branch point is the guanine. Okay. Not adenine. Or nucleotide cofactor GMP, GDP and GTP attacks the phosphate at the 5' splice site. Either this guanine will form a nucleophilic attack or GTP, GDP will form a nucleophilic attack on the 5' splice site. The 3' OH of the 5' exon becomes a nucleophile and the second trans esterification results in the joining of the to exon. You must be wondering what is transesterification. That is the bond which is broken down as a result of this splicing. This is known as the transesterification process. And this is the transistant intermediate, then this is the excised intron, and this is the ligated exon. The process is same, okay, whereas the certain differences like the G, here G is the branch point. Moving to the group 2, here the branch point is the adenine. This is the difference between group 1 and group 2. Group 1 guanine is the branch point and group 2 adenine is the branch point. Rest all the things will be same. Here again this OH will form a nucleophilic attack on this 5' splice site, as a result of which the free OH will be formed in the 5' end. This will again attack to the 3' splice site, and here a lariat formation will take place. Okay, this is the process which takes place in the group 2. Here the intron which is formed is in the lariat shape. One more important thing which I forgot to make you understood, that in this case, after in the pre-mRNA splicing technique, after the removal of the intron, what happens, a sequence will be present which will tell that there is a proper binding of the exons will be present. Okay, so that I have forgot to mention you, that I have mentioned. Apart from that, this is the 3 overall process which takes place in the splicing, that is pre-mRNA splicing. Here the lariat or the intron is in the lariat shape. Group 2, here adenine is the branch point and the intron which is formed is the lariat shaped, and group 1, here guanine is the lariat form and in linear intron will be formed. So this is all about the splicing technique. With this we come to the end of the process of transcription in detail. That is a process from, that is prokaryote, eukaryote and the modification purpose. Hope you understood this part, and if you have any doubt regarding this, you please do comment and share as much as you can among your friends. In the next part we will be dealing with the translation process, how translation takes place in prokaryotes, eukaryotes and rest of the things we will be dealing with. Thank you and have a nice day, and subscribe to my channel, like and share as much as you can, because these are very important. As I have already mentioned you, again and again I am telling you, these are very important. Share as much as you can, and thank you and have a nice day.